Alright, so today we're going to finish off the protein metabolism. We're going to talk a little bit about the modifications of the proteins. Over here we have an acetyl group being added to lysine and a monomethyl to lysine, dimethyl and a trimethyl to lysine. And more carbons you have, more hydrophobic it's going to be because it's nonpolar and doesn't want to interact with water, so this charge is being made less and it's going to become less polar. And it's important to know and memorize the acetyl structure. We know the methyl, it's just CH3. And the acylation interact it interacts with the DNA histone thing because you lose the charge, and the methylation is the interaction with the histone and other proteins. And only acylation removes the positive charge from the N. When you methylate it, it keeps the positive charge. And over here we have a hydrophobic chain being added to the amino acid. Over here we have glycine, and we have this mesteric acid being added so it can anchor into the cell membrane. The cell membrane, inside the cell membrane, you have those hydrophobic tails. Those are going to interact with the mesteric anchor here, and the palmitic group here, and the prenyl group over here. We have a cysteine here, cysteine amino acid. And then over here we have also a cysteine, and this one's the glycine over here. And we got hydrophobic fatty acids being added to the amino acid so that way they can anchor into the cell membrane. Oh, here it can be added to the N terminal or the O terminal. Over here it's being added to the S of the cysteine. The fatty acid is being added to the N and the S here. It'll also be added to the O. And yeah, and then over here, digestive enzymes, they need to be created in a non-active form, because if they were active inside the cell, they would just break down all proteins inside the cell. Over here we have trypsin being made. It's the zymogen form of the trypsin, and then chymotrypsin comes and um, activates trypsin once it's in the gut, where it's supposed to start um, carrying out its enzymatic activities. And then there's like another cleavage by chymotrypsin, and then it's still active. And then over here we have trypsinogen being activated by entropeptidase. And so while it's in the cell, it's a zymogen form or inactive enzyme. And then once it's in the gut, it's going to be the active form. And this one is about how our protein's going to be degraded. So usually first there's a phosphorylation, and then you have a polyubiquitin chain added to the to the protein. Here, polyubiquitin chain is added to a lysine residue, so that's the lysine and the polyubiquitin chain being added. Um, oh, here's the protein. The ubiquitin molecules are being added, and then they're sent to this cylindrical ATP-dependent protease structure, and the protein goes in there, and then it's broken down all the way down to its amino acid thing. So basically there's three steps. You got the phosphorylation. After the phosphorylation, you have the polyubiquitin chain being added to the protein on the lysine residue, and then it goes into the proteasome, where a ATP-dependent cylindrical protease is going to break down the protein down to its amino acid level. And And then it also unfolds a protein down to its primary structure, so that way it can rapidly degrade it down to the amino acid level. And this picture shows the same thing going on in a different diagram. So you got the protein coming in that's bearing the polyubiquitin molecules, goes in, gets unfolded, then broken down to the amino acid level. Here's a cylindral protease right here, this yellow thing. And then there's 4 different things that can happen with proteins once they're synthesized. Once they're synthesized, they can fold without any help. They can fold correctly without any help, or they can fold correctly with the help of molecular chaperone. Either the either the we had 2 different chaperone proteins that helped it fold, the HSP70, the heat shock protein 70, or the HSP60. HSP70 was the one that was like individual things, and then individual circle oval things, proteins that help it fold, and then there was the HSP60, which was like a chamber, and inside there they got folded. And or else, if it's not folded correctly and it's not broken down by the proteasome, then you have a protein aggregate like in the mad cow disease. Alright, so there's many diseases that are caused by proteases and protein aggregates. So one disease is sickle cell anemia, and these are often dominant disease because as soon as you have a gene for that, and it's gonna make the protein, and the protein is gonna create aggregates. Brain is especially sensitive to this protein aggregates because the cell can't just apoptose and get rid of the protein aggregate. The neurons, they don't replace, so they just die. And it also plays a role in Huntington's disease and Alzheimer's, where the protein aggregate blocks the axon in Alzheimer's. And over here, the changes in the secondary structure play an important role in resistance to proteolysis. Proteolysis is basically breaking down protein. Um, and then so there's a different beta structure which messes up the protein folding, and this is a normal one with the 4 alpha helices, and over here, I mean the 4 alpha helices, and then here we have 2 alpha helices and 2 beta sheets, the anti-parallel ones. And so this anti-parallel is going to make the protein sticky, and then so that's a normal protein, and there's a sticky one. Then this is very rare, and then once it becomes sticky, it makes more of these proteins into sticky ones. Then you got an aggregate, and there's 3 different diseases that that occur with this mechanism. You have the Creutzfeldt-Jakob disease in humans, and the bovine spongiform encephalopathy, or BSE. This disease occurs in cattle or scrapie in sheep. That's about it.